All right, so this lesson is all about DNA replication. You already know a little bit about DNA replication because we talked about it in the last chapter. This lesson is more about the specific, molecular things that are happening while DNA replication occurs. So on your handout, you have that picture on the side. There are a couple of things about DNA replication that you would need to know. The first thing that this picture is trying to illustrate is that DNA replication is what we call semi-conservative. So when you look at the word semi-conservative, semi means like not completely, partially. In this case, it'll be half. Conservative is talking about whether or not when we're replicating DNA, we have the DNA that was already there or whether it's new DNA being added. So when we say that it's semi-conservative, what we really mean is that each new DNA double helix has one old and one new strand. So on the picture in your handout, you can see a couple of things. On the top part of the picture, that's where you have the sort of same pictures that we had on the second page. So the same structures, the same symbols represent nucleotides and sugar and phosphate. So you can see these little round bits. Oh, my smartboard's off. Those represent the phosphate. You can see the little pentagon shapes. Those represent the sugar, so in this case, deoxyribose. And then you can see in the middle all of the different nitrogenous bases. Those three things, phosphate, sugar, and the base, make up a nucleotide. Now at the top, it looks much like what we talked about yesterday, where you have two anti-parallel strands. So you can see the phosphate and the sugar are sort of opposite orientation on either side. You can see that wherever I have C, G is always on the other side. Wherever I have A, there's always T on the other side. So you can see that it's complementary. But later on in the picture, starting about here, we see what we call unzipping. The DNA molecule is being pulled apart so that the outsides kind of stay together, but the middle is being pulled apart. When DNA replication happens, the two outside pieces are going to serve as like a template. They are going to be the old part. And in the middle, you can see there are new nucleotides being attached to either side. If this continued to happen, what you would see is that the entire double helix would get unzipped, and in the middle, on both sides, new nucleotides would be added. So the little picture on the bottom highlights that if I had replicated DNA, so now I have two double helixes instead of one, right? I replicated my DNA. The pieces on the outside, that would be the old DNA. And the pieces on the inside, oh, I wanted a different color. The pieces on the inside here, these would be the new strands. So if we compared that little picture to the bigger version, the part that I highlighted green would be all of these pieces in the middle, and the part that I highlighted blue would be these segments along the outside. So the picture is trying to illustrate, with the two little versions of it, that when DNA replicates, it has to unzip, and the new nucleotides will come in the middle. Some of them will attach to one of the original strands, some of them will attach to the other, so that when I have two strands now instead of one, so I've replicated my DNA, one side of it is always the original strand, and one side is always new. This, hopefully, will prevent most errors. Not all errors happen, but because we're not making new DNA from scratch, but we're actually using the existing DNA as a template. It means that there shouldn't be a lot of mistakes in terms of having the correct nucleotide in the correct spot. Now, you should already know, so I'm just reminding you of this, that this happens during the S phase of interphase. It would be a realistic thing, once we've done both of these chapters, for me to combine asking you about the cell cycle and maybe something about the molecular basis of replication at the same time, because now we know about both. And on the diploma, they'll regularly do that. If two things could be talked about at the same time, even if they came from different chapters or different units, that will sometimes happen. Now, if you colored the second page, it's the same symbols on this page, so we don't have like a new legend. I mean, there is one on that page. You could color it differently if you want to, but the shapes are the same as they were on the first page. The other thing that I wanted to note on this picture is that these are free nucleotides. Sorry, my smartboard is not catching my pen there. The place that you get the new nucleotides to add to either side is that your body has a bunch of them free. It could be from having broken down some cells that were damaged, and so the DNA gets recycled. It could be contained in the food that you ate. So if you ate food that was something that was alive, you ate some of its DNA, and so your body can break down that and recycle the nucleotides. So that you know, as far as we can tell, living organisms all have the same nucleotides. The way they're arranged is differently. But regardless of what you are eating, there would be nucleotides that your body could use to construct copies of your DNA. Now the other picture, and this is a picture if you ever needed it again, it's in your textbook too. This picture is going to help illustrate some of the molecules that are necessary for DNA replication to happen. On this picture, instead of representing individual nucleotides, we're just representing strands of DNA. So it's a little less specific than the picture on the top, but there are lots of other things that we need to label. When you look at DNA and it's replicating, you'll always see at some point the two strands splitting open and forming what is called the replication bubble. It looks like a bubble. It's the spot where the two original strands of DNA start becoming unzipped. So these are my original DNA strands. Now in my picture, they're kind of a darker blue and kind of a lighter blue, but you might want to highlight them with a different color so that it's clear, but it's the ones that are on the outside. There will be a point where an enzyme will come along and help those original strands to unzip. So where you see number 1 here, this is called helicase. It's an enzyme. If I didn't know that, I would look at its name and be like, oh, it ends with ASE. That's how enzymes end. And that could hopefully jog your memory as to what it is. So the job of this first enzyme is to unzip the double helix. Now, how does it do that? Well, you have to think about how the two sides were attached. When you built the model yesterday, if you got the two sides going, the thing that was attaching the two sides were those big white bendy pieces, which represented hydrogen bonds. So helicase can help break those hydrogen bonds, because remember, they're just a force that's attracting the two sides together. So if we just get rid of that force or break that force, then the two sides can come apart. So that's the first thing that needs to happen. We need to pull the original two strands apart and make a replication bubble. Then you will see a whole bunch of other enzymes come along, and different things happen on the top and on the bottom. Where you see number 7 pointing to this strand, this is called the leading strand. There are a couple of ways that you could recognize the leading strand. The most obvious way would be that it is continuous. So if you look at that strand, there's no spot where there's a break in the line. The other thing that you could use to identify the leading strand is that it goes in the same direction as helicase is moving. So on this picture, I put an arrow so that we would know this enzyme is moving in this direction. And so it would continue moving towards the left, pulling the two strands apart. You can always tell, even if there's no arrow, where the bubble is, the other way will always be the direction that that enzyme is moving. So the same direction that everything is moving, that is the leading strand. The leading strand represents new nucleotides being attached. Now, we didn't show individual nucleotides on this picture, but if I were to imagine what this looked like, if on my original DNA strand there was an A, there would be a T nucleotide here. If there was a C, there would be a G. If there was a T, there would be an A. So the new strand, that's called the leading strand, would be formed complementary to the original DNA strand. Now, the new nucleotides don't just show up out of nowhere. An enzyme has to help them so that they know where they need to be attached. So you will see all over the place, here, 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 a whole bunch of copies of enzyme number 2. Enzyme number 2 is called DNA polymerase. You can see that it ends with ASE, so it's an enzyme, just like helicase is. If you look at the name, poly means many. So this is the enzyme that is going to bring many pieces of DNA so that I can make a new strand. Now, so far, does anyone want to ask any questions about what is happening when the DNA is replicating itself? Okay. So that happens on the leading strand. Sometimes, not all the time, but there is on this picture, you will see the numbers. I would have mentioned them yesterday when I talked about things being anti-parallel. DNA always has a 5 and a 3 prime end. Replication always goes from 5 to 3. Okay? Now, I'm only mentioning that because it's possible that you could use that as a clue if you were ever looking at a picture and you knew, oh, I have to start on the end that says 5. Not every picture is going to have the 5 and the 3 end labeled. Those numbers tell us where the sugar and the phosphate are. So the 5 prime end is the end where the phosphate is on the end, and the 3 prime end is where the sugar is on the end. Now, the other strand is a whole lot different than the first. There are spaces in it, and because of that, where you see number 8 here, this is called the lagging strand. Clearly, the same thing is not happening. It's in little bits and pieces. There are extra enzymes all over the place. Things look a lot different. The lagging strand is not continuous. Okay, so the word continuous means that there's no break in it in this sense, so we can clearly see there are breaks in this strand. Because DNA has to go from 5 prime to 3 prime, it can't start right here. That's the 3 end. It has to start over here. So if you look at these arrows, they're showing you that the lagging strand is not moving in the same direction as the top one. So it's going in the opposite direction, but it's still 5' to 3'. Because the two strands of DNA are anti-parallel, 5 to 3 on one side will be the opposite of 5 to 3 on the other side. Now because of this, and so that DNA can get replicated kind of at the same pace on both sides, instead of replicating one long strand and having that strand move one way and the leading strand move the other way, little tiny fragments are replicated kind of all at the same time. So each of these little tiny pieces here that you see in the lagging strand are called Okazaki fragments. So if you're ever a scientist, name stuff after yourself, and people will forever have to refer to things as like the Melissa fragment. I wish I could name something after myself. There was a computer virus several years ago called the Melissa virus. It wasn't awesome, but I was like, wow, that's so cool that it's named after me. Not me personally. Anyways, these little fragments don't start the same way. One of the things that you'll see wherever you see number 4 is an RNA primer. If you think about what primer is when you're painting, primer is what you put on the wall first so that the paint sticks better. The RNA primer, in this sense, is what we're going to bring first so that the DNA has something to stick to. So the same way primer works for paint, this little bit is a place for the DNA to start attaching itself. The primer doesn't show up all by itself. Number 3 is another enzyme called primase, and its job is to attach the primer. The primer's job is to provide a place for DNA to attach. So at the beginning of each of those fragments, there will be a little primer, and an enzyme called primase causes that primer to be attached. Now, just like on the leading strand, you will see number 2, right? The same thing that it was up here, DNA polymerase. DNA polymerase's job, just like it was on the leading strand, is to bring many DNA nucleotides. I'll just write that part down. So on both strands, DNA polymerase is acting. Notice that there are several copies of it and that they are both on the lagging strand at different spots at the same time. So these are not distinct processes where first one thing happens, then the other. This is all happening all at once, all at the same time. The other enzyme that we need, number 5 down here, is called ligase. I was watching, what was I watching last night? Riverdale. Oh my gosh, how did I not watch that before? I watched the first 3 episodes and I made myself go to bed. But in the first episode, or maybe the second one, they find someone with ligature marks on their arm. So ligature is like when you tie something up. Ligase here, coming from the same root, is going to attach the Okazaki fragments together. So a lot more stuff needs to happen on the lagging strand as compared to the leading strand. Now, does anyone want to ask any questions about anything that is on this diagram? Yes. The Okazaki fragments are the little pieces, each of these little blue segments that make up the lagging strand. So the lagging strand would be this whole section. Each little piece would be an Okazaki fragment. Does anyone else want to ask anything about what's on the diagram? Okay. So